what I'm going to talk about today are sort of the trials and tribulations of moving your research program into something that's a little out of the box, an opportunity that I had to do, I had a number of years ago to go through it. And one of the questions that people ask me is, how did you end up doing this? Aren't you like the looking time guy? And the answer to that is yes, I am. This is sort of a quick map of our research programs at the University of Kansas. This is one part, and the part I'm going to be talking about today is this one, where we're looking at attention as an outcome in early interventions. And effectively, the intervention is the development of, or is our nutritional manipulations and clinical trials involving nutritional manipulations. But it all springs from a basic program of research on development of attention. We spend a lot of time trying to figure out really what attention is, how it develops, how you measure it, and all that. And I'll try to talk about how we got here as part of the talk. And actually, that's the first piece of it. We measure visual attention by using an old paradigm called visual habituation. And it's a slightly sneaky way of doing it. Essentially, you sit a baby in a car seat, and you put a stimulus up and let them look at the stimulus, and you watch them while they watch the stimulus. And you code their looks over time, and you present the stimulus over and over again, and you'll see a film of this. And while they are looking, you're actually also measuring their heart rate. And we started doing this about 25 years ago. And based on the work of our friend John Richards at the University of South Carolina. And what we do really is measure heart rate, because as babies are looking, you get a really nice parasympathetic response. Baby's heart rate drops. And John's shown without a shadow of a doubt that a lot of the basic fundamental cognitive activity that goes on while babies are looking occurs while the baby is in that phase of sustained attention. That decelerative response, that decelerative phase. And so keep this in mind. We'll see this figure a couple times today, but the SA, the sustained attention, is the piece that really reflects when babies are looking, and when babies are actually attending and engaged in information processing. Orienting is sort of the part where they're getting oriented toward looking at the stimulus. And attention termination is a period where you're looking but not really paying attention. We call this being a teenager in the lab, and some of you might be aware of that. This is what one of our basic paradigms look like. If I can get it to run. And this is what it looks like. It's a dark room. You present a stimulus. The baby's looking at it. He stopped on his way to the prom. You can tell by his hair. And you can see this first look is pretty long. He's got to look away for a second. And he'll sort of show little glances, but you can see the stimulus is right in the middle of his eye, and that's what we use. And eventually he'll look away for more than a second. We'll turn it off and turn it back on again. And you'll notice again he's back up there. And he's looking, but now what you'll notice is that he'll look for less. He'll look for less time. And we'll do it a couple more times. Sometimes you get a little recovery of looking. I think you'll get a little bump here. But what's happening is, as the same stimulus is repeatedly shown, the baby habituates. And you can see here, he's done. He's like, are you going to show me something else? Let's go. And eventually, he'll look for a really short period, and then that's pretty much it. And there's actually a calculation of an algorithm, and then we give him a choice trial between looking at what he's seen before and a novel stimulus. And then you can see that's why he's shifting back and forth, and we have a choice trial. This is a recognition memory test that I'm not actually going to be talking about today, but it's the kind of thing that we do. And this is the basic paradigm, just so you know. What we did for the first 20 years of the work that we performed on this is look at individual differences. People use the habituation as a tool to get at what babies can do or can't do at different ages. For me, this was a measure of learning. And what I really wanted to do was use it as a measure that could be useful either as an indicator of individual differences that might be predictive of how kids turned out later on, or maybe as a way for measuring, at least in the short term, how kids were functioning, maybe as a function of things, interventions, or whatever. Through the 1980s, we did a lot of work on the psychometrics. We wanted to know what the short-term reliability of these measures were, what the long, sort of the stability of these measures were over time. The next 10 years we spent working on how individual differences on those measures related to performance on different cognitive tasks. And then we did a large longitudinal study from during the 1990s and into the early 2000s on the predictive validity of these measures. And what we basically found was, and all the references are there if you're interested in looking them up. Basically, and what we found was that as babies got older, the amount of time they spent looking dropped, and the amount of time that they spent in these sustained attention and attention termination and orienting varied across time. But to kind of get down to where, to sort of the most succinct way to say it, is babies who show a clear and precipitous drop in looking time across the first year, and where they maintain a proportion of sustained attention well during the first year, tend to be associated with the best outcomes. And to kind of give you a picture of this, this is the results of, and I've very much simplified them, and from our longitudinal study. The top group are basically the measures, what the measures look like for a group that scores very high on an IQ test at 4 years of age. So basically you see a drop in looking time, that's on the left here in black. And then what you see is the top gold line is sustained attention. You see basically it's flat across the first year. What we found for groups that were scored less well on those outcomes, that basically the drop in looking time was less severe, but also that there was a drop in sustained attention over time. So just keep this in mind, because it's a way that we use to evaluate whether children are doing okay or not okay or whatever, as a function of the kinds of things that we use in the way of nutritional manipulation. So this is the groundwork, in many ways the grunt work that got done in order for us to be able to make the next translational leap. So in 2000, so this is where I've been asked about 4 times today. So how did you end up doing this? And this is where it gets interesting. People had started to incorporate, without my knowledge, a lot of these measures. They had gone out and they actually read this stuff. They started to incorporate our measures into some longitudinal studies where they had provided some nutrients to babies. And at this time there was a big, at that time there was a very large controversy over whether or not the addition of these lipids into formulas was going to be a positive thing. And I started to get phone calls from scientists saying, hey, we tested whether babies who were given these lipids were doing better, or we followed them up on your tests, and we were told by somebody else in the field that we should call you because you will know what this means. And so we started to, and it turned out that a lot of the measures were showing that babies were doing better if they were supplemented, particularly preterm infants. They were showing shorter looking times, and they didn't quite have heart rate at that time, but shorter looking times. And eventually what happened was that one of these people, who was always calling me on the phone, ended up moving to Kansas and moved to the KU Medical Center, and that's Sue Carlson, who's been my longtime collaborator since she moved in there. And we just decided that we would build a lab together. And so we invested time and some resources. We were given space by the then IDRIC director, who now works for me, and we sort of started on this sort of long, happy trail together. And one of the first things we worked on was a prenatal supplement study where Susan fed moms high DHA eggs. You can get DHA into your diet if, for example, the chickens are fed algae, because that's where the DHA comes from. And DHA is docosahexaenoic acid. It's one of the omega-3 long-chain polyunsaturated fatty acids, which Susan pronounced perfectly. It's an LC-PUFA, and we'll talk about that in a minute and what it means. But basically here, what she found was that if you fed moms these high DHA eggs, their pregnancies were longer, were by a week. So, and that was important because the suggestion there was it might prevent prematurity. Well, she then said, well, why don't you follow this cohort? And I said, okay, we'll put them into one of our paradigms. And what we found also basically here was that we were able, it wasn't necessarily the actual clinical trial didn't work, but divided moms into high and low DHA based on what they had eaten during pregnancy. And, and what we found is that kids from moms, babies from moms with high DHA actually showed shorter looking at 4 and 6 months of age on this paradigm. And then when we followed them up with a free play attention task that actually was developed in Lisa Oaks' lab while she was in Iowa, we found that basically those babies were also far less distractible at ages 12 and 18 months of age. So again, I remember the phone call calling Susan and saying, are you sitting down? Because there's a big effect here that we're getting as a function of the high and low DHA levels of your kids. So of the kids in this study. So it started this long collaboration, and there's Susan in the dark shirt there, and then this is our team, our core team. That's Byron Gajewski on the right as our statistician, Susan Carlson, Kathleen Gustafson's our electrophysiologist, Beth Curling, Sarah McCone, who's our neuropsychologist, Jocelyn Thodosoff, and then Jill Shaddy, who was also one of Lisa's students back at Iowa. So there's quite a bit of crosstalk here. And what I'm going to show you here are the effects of 3 clinical trials, 1 postnatal with LC-PUFAs, 2 prenatal trials, and the last one's pretty interesting. But to start off, I'm going to show you the results of an NICHD study that we did in Peru to just kind of show you how this works. This was a study of zinc, and it's nice because I have a travel log of it. I have pictures. This is the team, and actually there on the steps on the second person from the bottom is Kathleen Canis, who is Lisa's old Ph.D., not old, but former. But that's on the right bottom is Laura Caulfield, who is a professor of international nutrition at Johns Hopkins. We worked in Peru together, and that's a basic Peruvian infant there. You got your basic model there. And what we did is we went into a population outside of Lima, and Lima is an interesting place. It's a desert, and it's a very poor city. And we worked out of this clinic, and for $8,000, we were able to build the addition at the top of this building there, which was amazing. I don't think you could do that here in the U.S. And then across the street from that building, if you're up in those windows, you're looking down. This is the clinic, the pharmacy, and we were able to set up a longitudinal study, a supplementation study with infants, because these infants all went, because of their diet, they all went anemic by the time they were 2. And the reason for that is that they don't eat much meat. This is a picture of the area right outside the clinic where I just showed you. And you see that hill in the back. It's just completely covered with shanty town cardboard and corrugated steel houses. And about 500,000 people live in this area. It's called Villa El Salvador, outside of Lima. Just to the right, actually, there's a cliff, and then there's a Pacific Ocean. But these folks don't eat much. They eat potatoes. They eat some vegetables. They don't eat fish, which is kind of strange, and they don't eat any meat. And so what happens is they don't get much iron, and they don't get much copper, and they don't get much zinc. So what we do is go in and we address the anemia by giving babies who are born to moms in this clinic iron and copper, and then everybody gets iron and copper. So the idea is we're going to eliminate the anemia by doing this, but then for half the group we randomly assign them to also get zinc, because there was an opportunity to look at this. Now in the past, zinc has been associated with improved motor development in babies, but what we did is essentially start them on at 6 months of age and then follow them through 24 months, again on sort of habituation and free play outcomes. This is the data from the visual habituation. The zinc group shows a very clear decline in looking time over age. Again, 6 months is the start of the supplementation. And what you find is that this group here, who's getting zinc, the solid line, they drop over time, as they should. Whereas the group not getting zinc actually is flat, which should not happen. That's sort of an aberrant pattern, an aberrant developmental trajectory for this measure. And they're different by the time they're 12 months of age, which is exactly what predicted. As we followed them up at 12 and 18 months, we also found that these babies were more active in free play, suggesting that, again, the motoric aspects of, and this is just the number of shifts they make between targets in a free play situation. We give them a toy, and we code how many times they switch their attention between that. And at 12 months, they did more of that, suggesting that they were just more active over time. Interestingly enough, the physiology of the attention wasn't affected by this, but more just the motoric aspects of looking and moving around from one stimulus to another. So again, another positive outcome with respect to the kind of nutritional supplements that we were doing. Interestingly enough, we administered the Bailey Scales of Infant Development, which is a standard global developmental measure of developmental status. It's meant to detect whether or not babies are doing okay or not in a very global way, and it's constructed like an IQ test. And although I didn't break it out by groups here, this is basically just to show you that this population was attaining normal scores, and nowhere did we see any effect of the supplementation on the Bailey scales. And this is going to be an important point because this is a global developmental scale. It should have come out, but we are seeing the effects on these other more granular, more high-tech measures of attention, but not necessarily on these global scales, suggesting that maybe those global developmental scales are not as sensitive to these kinds of effects as we might hope they would be. Now I'm going to get into the long-chain polyunsaturated fatty acid, or the LC-PUFA literature. There are two that we have been working with. One is arachidonic acid, that's the one on the left. Docosahexaenoic acid is the one on the right. And you have the omega-6 and the omega-3 family. In true American fashion, they are labeled bad and good because omega-6s, we have a lot of those in our diet. We get it from dairy, eggs, meat. And it does have some positive effects. And the idea, at least in some areas, is that we should reduce omega-6s and be high in omega-3s. We actually need them both. The omega-3s are really important, and we think we're especially interested in docosahexaenoic acid, or DHA, because it's an essential omega-3. And what's really interesting is you can actually, hardly anyone is deficient in ARA, but a lot of people are deficient in the United States with DHA because there's only one way to get it, because we don't make it from precursors in our diet, but we have to get it preformed for the most part in our diet, and the best way to get that is by eating cold water, oily fish, which Americans, for the most part, don't do, or they do it in very low amounts. And you can see that DHA has a whole bunch of really good things. It ends up in the brain, it ends up in the retina, it promotes neural transmission, and it seems to be involved in the catecholamine synthesis. So it has a lot of things that could happen. But the big reason that we're interested in it is because it accumulates in the brain over development. And so here's post-conceptional weeks, and so we're out, and the red part is prenatal, the gold part is postnatal. But you can see that DHA accumulates in the brain very rapidly during early development. This is not a profile that you see for example, ARA. It's just flat, and again, people are typically not deficient in it. So the idea here was that this accumulates, you have to get it from your diet, it accumulates, it's developmentally relevant, it accumulates in your brain over time, or especially early, and then it starts to plateau after this. And so it might be important in cognition. So we started working on whether or not, how much of this should we have in infant formula. And one of the studies we ran was the Diamond trial. I'm just going to kind of show you the basic design. The idea here was that it's a dose response curve design looking at DHA, although there was an introduction of ARA above the placebo. So the placebo formula was 0 and 0, but the company introduced, this study is funded by Mead Johnson Nutrition, and 64.64% of arachidonic acid, and then 0.32, 0.64, and 0.96 percent of DHA. And we fed babies, we randomized them to these 4 groups, fed them formula for 12 months, moms got the formulas for free. And we know that by giving them these different formulas, by taking blood measures of DHA in in their blood, we know that it differentially affected how much DHA was in their systems. This is the consort diagram to just show you what happened to all the subjects. We randomized 160 kids thinking we'd have 40 in each group. By the time we finished, the original study was powered to get to 18 months. We had between 20 and 30 kids at that point per group. And then we continued to follow them, and at least we kept about 20 kids per group by the time we got out to age 6. So I'll show you some of the findings coming out of this. One thing that you need to know before you go into the findings is the fact that this is a very poor sample, a very low SES sample, typically inner city, low SES. You can see here that I'll point out a couple things. The maternal education is quite low. They're basically high school graduates. And none of these variables varied as a function of the groups up at the top there. The household income was about $34,000, $35,000. And then here's a really killer variable, which is parental PPVT. And you can see that it is not particularly optimal. Again, that's a scale that runs from where 100 is the average. So these parents are about a standard deviation below the average for that. So that's the parent measure of sort of verbal IQ. Here's the longitudinal schedule that we had going for them. Here's when we measured all things. We measured visual acuity, we did visual habituation, we did the Bailey scales, we did dimensional card sorts, Stroop task, we did PPVT, Tower of Hanoi, electrophysiology, and then the Wechsler at different ages. And then you can see there's some overlap in the design. To give you an idea of that I'm not cherry picking results, every place there's a green check, we saw a positive effect of the formula we were feeding on these kids. And it's important to note that we stopped feeding this at 12 months of age. So anything that happens from here over is actually an effect that's persisting despite the fact that we have not added anything. We have not added any more LC-PUFA to their diet. The first thing that we got at 4, 6, and 9 months was the fact that we improved the quality of these babies' attention. This is the percent of time they spend in SA, in sustained attention. Remember, it's that decelerative phase that I showed you early on. And in the 2 middle doses, we found that they had more time spent in sustained, or a higher proportion of time spent in sustained attention than the control group. As you can see, what's really interesting here in this highest dose here, which is the 0.96 group, is that we didn't see, that wasn't significantly different from the control. So there seemed to be a reduction in benefit. We thought initially this would be some sort of anomaly, but it turned out to be something that carried through for the most part for the rest of the study. If you look at the change in sustained attention over time, what you find is at the supplemented group didn't change. That's the gold line over time. This is a plot of the proportion of sustained attention as a function of age. The gold line is pretty much flat actually statistically, whereas the unsupplemented group dropped their looking time. And if you remember back to the data that I showed you from the normative groups that we showed, that presages the drop in the blue line presages a worse, presumably worse developmental outcome. We did the Bailey again, and guess what? Nothing happened. Again, the assessment of the Bailey scales at 18 months of age was not sensitive to any of the groups, and certainly didn't show up. And this is important because a lot of the data, if you start reading, for example, meta-analyses, they'll use the Bailey scales to try to evaluate the effects of LC-PUFAs in infant diets, and what you're finding for the most part is that there's no effect. Well, we found that too, but we did find, for example, it improved baby's vision, it improved baby's attention, and what's really interesting is well after when we stopped feeding them the formula, we continued to see positive effects. One of our favorite tasks is the dimensional card sort. We did this at 3 years, 3.5 years, 4 years, 4.5 years, and at 5. So we measured it a bunch of times over the preschool period, and it's a great task. And some of you know what this is already. You present them with, and it's all actually, there are no boxes and no cards. It's all computerized, but this is easier to present. You present kids with, for example, a picture that can be sorted on one of two dimensions. So you can take the blue bunny there, and you can sort it based on color. So in which case, it would be sorted with the blue car, or it can be sorted based on shape, in which case it would be sorted with the red bunny. And so you can sort on shape, or you can sort on color. And then what you do is you train kids to do this and teach them this rule, and then you say, okay, after they've mastered it, we're going to change the rule, and now we're going to switch it so that if you were trained on color, we'll change it to shape, and if you trained on shape, we're going to change it to color. And what happens is that we get an overall score of both that incorporates both how well they learned the rule in the first place and also how well they adapted to the change in the contingency. This is the control group. And what you can see at 3, 3.5, 4, and 5 years of age is that the kids in the control group didn't really get better at this task until they were 5 years of age. So we administered it over and over to them, and they really didn't improve until about 5 years of age. The 2 middle doses, what we saw was that they actually showed a very clear progression as they got older, but they were different from the control group by the time they were 4. By the time they were 5, they were pretty close, but they were still ahead. And so basically what we've done is accelerate development by a whole year, right? At 4 years, this is 4 years of age, this is 5. So we basically have accelerated the development of their cognitive function by a whole year at this time. And then here is the highest dose. And again, interestingly enough, it's an intermediate effect. It's a clear reduction of benefit based on raising the DHA to that level. By the time these kids were 5, we administered the PPVT. And again, we find that the same basic pattern. Here, the highest dose is almost statistically significant from controls. But basically, here you can see 0.32 and the 0.64 group is significantly above controls. Notice the reviewers all asked us, how do you get a group of kids that score 85 on the Bailey? On the PPVT. And we just, at that point, were able to say, remember what their parents scored. And if you remember back to that, remember their parents are scoring only about 85. This is exactly what we'd expect to have happen. And if you're worried, we covaried parental PPVT in this analysis anyway. So again, we're finding that there's a persistent effect. And remember, I'll say this one more time. This will be the last time I say this. We stopped feeding them at 12 months. So this effect is persevering over 4 years later. We did we started to do the Weschlers at 6 years of age. We ended up, there's only a marginal effect on verbal IQ, although you can still see the pattern through all of this, where the 3 supplemented groups are actually higher than the control group, which is the white bar. And if you combine those or you collapse those 3 groups, you do find that the supplementation had a significant effect on verbal IQ and a marginal effect on but you can see the effect is not necessarily as strong. So in any case, we are 5 years out from the end of, or 4 years out from the end of feeding. And we are still seeing, pretty interesting effects on IQ. At 5.5, we put them through an electrophysiology battery. We gave them what's called a go-no-go task and measured the electrical potentials from their scalp using ERPs. We actually didn't use a cap. A lot of these children were African American, so we actually had to use a 1020 system and apply the electrodes directly to their heads. And so our team is just great to be able to do that. This was the task, basically. They were taught, they were presented, sat in front of a computer screen. They're presented with fish, and they were told, every time you see a fish, hit the space bar. That's a go. But don't press if you see a shark. And so they would have to press if they see a shark. They had to inhibit that response. And so what we're really interested in is the difference between their brain responses to go and no go. They were actually the 3 supplemented groups were marginally faster. So this is important because they weren't any more or less accurate than the control group, but the 3 groups here that were supplemented here over at this point, they were marginally faster overall than the controls. And we measured the N2, which is generally, there's all sorts of, I'm nervous talking about this with Steve here, but it's basically an attentional indicator. It's the negative component at about 200 milliseconds, between 200 and 300 milliseconds, and depending on who you talk to, it has all these things, including inhibition, attentional shifting, and what we're interested in is whether this particular component was different as a function of what the children were fed, and it turns out that this is the difference between go and no go. The closer the lines, the squiggly lines are to 0, means that the response was the same in the go and no go situation. And so what you have here is that the control group basically is not different during the N2 period from 0, but that the supplemented group actually seemed to notice the difference on the no-go trials. So that they actually seem to have a brain response that was involved in inhibition. The topography over the scalp of the responses are pretty interesting. This is at 300 milliseconds, and as you watch the brain change, the right side on the right is the LC-PUFA group. You can see that there's a very clear activation that extends up to the parietal and maybe into quasi-frontal. So I'll run it backwards again. But here's 300 milliseconds, 400 milliseconds, and look at the right side, 500 milliseconds, and then back to 600 milliseconds. So clearly the formula has changed the topography of the electrophysiology over the scalp. So that's where we are right now. We're still following this group. We're bringing them in for imaging, but we're getting down to where there's not that many kids left in the group because we've just lost track of them and it's a small group. But we've got plenty of other stuff to talk about. That was a postnatal trial. We fed postnatal. We fed children. We fed those infants with formula postnatally. This is a prenatal supplement, and remember that the slope of that DHA accumulation is pretty steep during the prenatal period. We thought that actually, if you provide babies or provide moms with DHA early, then it might have an effect. And the HOPE trial was a very small R21 that we ran, and it was based on the idea that that DHA might be affecting the integrity of the autonomic system. And we were really interested in this, and there's a paper in PLEFA, and here as a psychologist, here you are. I never thought I'd publish in prostaglandins, leukotrienes, and essential fatty acids while I was in graduate school, but here I started to have a lot of papers in that journal. But the idea here is that it's mediated by the effects of DHA are mediated by the autonomic nervous system. And so what we did is we enrolled moms at 12 to 20 weeks of gestation and randomized them to either get 600 milligrams of DHA or 0. They had corn oil. We took bloods. We know that the supplementation was bioactive. And our primary outcomes were fetal heart rate and fetal heart rate variability. And our secondary outcomes were a neonatal assessment called the Neonatal Behavioral Assessment Scale. We sometimes know this as the Brazelton scale. This is what we use to measure fetal heart rate. It's a fetal MEG. It is an instrument designed to take brain measurements from a fetus. It seems to be poorly designed because they didn't seem to realize the fetus moves around. And so what we use it for, and what it's really good for, is measuring heart rate, because you can get the fetus's heart rate, you can get the mother's heart rate, and you can subtract the signals and also subtract maternal breathing and fetal breathing. You get a really clean heart rate response out of the fetus. Again, we randomized about 70 moms to the placebo and DHA, and we found that by the time we got them to delivery, we had about 44 kids still in the study, but we measured them all along the way. I won't bore you with that, but there were no differences between the groups on any of our ancillary demographics, except for the fact that they had higher DHA in their blood, and that makes sense because they were given DHA. Fetal heart rate variability was higher in the DHA group, in fetuses whose moms were fed DHA. We saw a difference in fetal heart rate. We would expect that the DHA would lower heart rate, and we saw that trend, but it wasn't statistically significant. But we did see a very big difference, and heart rate variability is sort of a measure of does your biological system respond to different events? So a person with high heart rate variability would be sitting in a room and would be experiencing different things and having their heart rate change as a function of the things they were experiencing, as opposed to someone who had very low variability, whose heart rate would be sort of stable across any event or any stimulus that was presented. And that variability actually is actually claimed to be quite a very predictive and positive indicator from the standpoint of autonomic function and indicates the integrity of the central nervous system. We've improved that experimentally by giving moms prenatally DHA. When babies were born, we administered the Brazelton scale to them, and we found that, interestingly enough, babies who were from moms who were supplemented were marginally different in orienting. This is a measure of attention, measured in sort of a rating scale that you do with the baby during the neonatal period. They were significantly better on motor maturity, and they were significantly better on an autonomic scale, which is exactly what we predicted, which measures sort of the general reactivity of the response to different environmental challenges and stresses. So this is all in line with what we expect to see. The last study I'll present is the Kudos trial, and if you notice, you got to find an acronym for all this stuff, right? You just can't, our team spends a lot of time doing this. That and the color of the binders, where I really give them a hard time. But the Kudos trial is a DHA outcome study, and this is funded by NICHD. And this builds on the previous work that I showed you, suggesting that DHA actually increases the length of gestation. We thought, well, let's see, does it? If that's the case, what we might be able to see is whether or not DHA would actually prevent prematurity. And so the trial is a very large trial. It was funded about 10 years ago, and we're now in our last year of it. And we randomized a large sample of moms. We started with 350 moms, enrolled them, and we randomized them to either 600 milligrams of DHA daily or zero. So basically, they got 600 milligrams a day or nothing. And we, again, took bloods, and we know that both the moms and the babies after they were born were higher in DHA than if they were in the supplementation group. We were interested in a bunch of different pregnancy outcomes for the most part. Gestational age, birth weight, birth length, those are the primary outcomes. And our secondary outcomes were, again, sort of head circumference, prematurity, very low birth weight, or early, and hospitalization. This is the consort diagram. Again, 350 moms randomized to either placebo or 600 milligrams of DHA. And by the time we were shooting to have 300 by the time we finished, and we were quite close to it. And we're proud of this because about 80% of clinical trials actually fail to meet enrollment. And we were really proud of the fact that we actually hit, and we hit it on time. This is a much more educated and higher SDS sample than we saw in the postnatal feeding study. So that is important to bring to bear on this. But there were no differences on any of these variables as a function of group membership or randomization. To make a long story short, we found significant differences between the two groups on almost all of the pregnancy outcome variables. We saw higher gestational age, higher birth weights, longer birth lengths, bigger heads, a smaller percentage of birth weights under 2,500 grams, a significantly smaller percentage of birth weights under 1,500 grams. And those are the high-risk kids. We didn't affect prematurity, but we did affect high-risk prematurities, that is, prematurities under 34 weeks. We didn't affect whether babies were admitted to the NICU, but if they were admitted to the NICU, there was significant effects on how long they stayed, and if they were hospitalized, otherwise not in the NICU, if they were hospitalized, it was really a very set of interesting positive outcomes. And this is the selected outcomes. These were the birth weights. You can see basically we eliminated very low birth weights. There was no one in the sample that had that. We had very few early preterm births, and that was highly significant. And here are the hospitalization data. So this was really striking, set of outcomes for us to have. Basically, what we did was this. If this is the plot of the frequency of births as a function of weeks of gestation. So here's, on average, it's 39 weeks, somewhere in that range, and this is the frequency plot. And you can actually devise, and the University of North Carolina, they've done sort of cluster analyses and determined that this distribution can be divided into low-risk births, moderate-risk births, and high-risk births. And this is taking into account birth weight, birth length, gestation length, and a number of pregnancy variables. Our analyses suggest that we took the DHA supplementation of the moms took 40% of the kids from the high-risk births and moved them into the moderate-risk births. So this was a really striking finding that we were very, very proud of. One of the things we did was we economized this finding. What would it, just in terms of the hospital costs alone, how much does this save? And if I can read this last sentence down here, basically providing 600 milligrams of DHA for 26 weeks, and that there's a cost associated with that. The net savings per diet, mom and infant diet, was about $1,500. Extrapolating this to nearly 4 million US deliveries per year suggests universal supplementation with 600 milligrams a day during the last 2 trimesters of pregnancy could save the US healthcare system up to $6 billion. And this is, again, based on just our sample, and it's an extrapolation that our statisticians say we can actually talk about. So this is a really striking outcome. But it's a sample of 350. We only had a handful of these early preterm births. We're really concerned about reproducibility and reliability of our results. So how can we really be careful about this? We decided to get with our colleagues at the University of Adelaide, Bob Gibson and Maria Macrady's, who are also really extremely productive scientists in the area of LC-PUFAs and maternal and infant health. And they had run a sample of 1,100, a clinical trial involving 1,100 kids and moms in Australia. And what we basically did was say, we've got this effect, can we bring our statisticians together, so that's Lisa Yellen and Byron Gajewski, and can we combine our samples to see if we are getting the same effect, and do a Bayesian analysis in terms of the prediction. And basically, we found that we are getting the same size of the same effect size. And we were able to actually calculate, the statisticians were able to calculate that, that in the US, we would basically eliminate 106,000 early preterm births by simply using this. And in Australia, it's less because the population is less. Just to give you an idea, here's the plot of birth weight by gestational age for the Kansas sample. The blue dots, it doesn't show up here. The blue dots are placebos, and sort of the open green dots are DHA supplemented. And so here, along here, is where the preterm births are, and you notice that the predominance of dots there are blue. That is, they're placebo. So this is where the high DHA, the placebo kids are, and where the high risk pregnancies are. And you can see almost all of them from our sample are placebo kids. Here's the domino sample from Australia, and again, look at the tail. the tail of the distribution here is dominated by kids in the placebo group. So we were able to show by combining the samples that we're getting the same effect, that this appears to be, in fact, a uniform finding across samples across 2 continents. And this just came out the other day, so we're really happy about this outcome. Oh, and we followed these kids up, which is what we've been doing for the last 10 years. And what we're finding, and we just published this one, and it's coming out in Pediatrics Research, we obviously measured them on visual habituation. One of the interesting things we found was that, and we've had conversations today with all sorts of people about sort of anxiety and mood and social cognition and all sorts of stuff, that babies from moms who were supplemented actually made it through our attention tasks at a much higher rate. We found that our subject loss was highly attenuated in the supplemented group relative to the placebo group, particularly at the older ages, at 6 and 9 months, but it came out overall. And this is just a contingency table of fussy or not fussy as a function of whether they were in the placebo or supplemented groups, p-value, and then here's the effect size in terms of a contingency coefficient. Basically finding that babies were better tolerated the task. And what's important here is that we didn't include any early preterm babies in this. This is a pure effect of DHA, not mediated by prematurity. We also found that babies from moms who were supplemented didn't change. They're the gold line. Their percentage of sustained attention didn't change across the first year, whereas babies from moms who received placebos actually declined in their attention, in their sustained attention, percentage of sustained attention. And again, it's important that there are no early preterm births in this follow-up. So there's not any mixing of an indirect effect of the effect of, for example, DHA on prematurity as affecting attention. This is a pure effect. We're going back in. We have these kids measured out now to about age 6. I will say that our preliminary looks at this effect is that it doesn't persist the way that the postnatal feeding studies have persisted, but we still haven't really analyzed the whole thing, so I don't have all of the data. But at first glance, this effect seems to go away. And so it may be that this produces a transient difference that goes away later on, or that any effect that you see as a function of prenatal supplementation might be mediated by preterms. And so we have another grant that's under review in a couple of days in Washington to see if we can replicate that. Guess what? The Bailey didn't come out. So once again, as a function of these standardized tests, they're not sensitive to these effects on nutrition. So I've taken you through 4 studies right now, and they're all longitudinal studies assigning kids to randomized controlled clinical trials where we've assigned kids to different levels of nutrients. And what I just wanted to point out here is that in every case, we are seeing differences on these granular, experimental, basic science type of tasks, but not necessarily on these standardized global tasks of developmental status. So to summarize, I hope I've convinced you that early nutrition and nutrient status can be directly effective to improve cognitive function, especially early in life. That the effects can be seen either in terms of prenatal supplementation or postnatal supplementation. That many measures of cognitive development that were developed in laboratories like mine and Lisa's and Susan's over the years are sensitive to these effects, but that are missed by global standardized developmental scales. Remember that when you see the New York Times publish, for example, a meta-analysis showing that there's no effect of LC-PUFAs, and they're basing the meta-analysis on the use of Bailey scales, which tends to be how it goes down. In terms of specific findings, postnatal feeding with LC-PUFA enhanced formulas produced lasting and meaningful outcomes all the way out to well after this feeding was ended. Postnatal feeding with zinc in a deficient population restored developmental course of attention during infancy and a profile of higher motor activity during the second year. Prenatal supplementation of moms with DHA changed fetal cardiac, changed the autonomic system basically, and positively affected perinatal measures of neurodevelopment. And prenatal supplementation also improved pregnancy outcomes drastically and positively affected sustained attention during the first year. And those are coming from those specific outcomes. The last bit is sort of a set of meta conclusions. Our labs went from basic science to translational by basically being an integrative, collaborative team. A psychologist, that would be me, a nutritional biochemist, Susan, electrophysiologist is Kathleen, and a biostatistician with expertise in clinical trials. And all of us have very clear roles and very clear responsibilities in this team effort, and we've got a great staff, and we've trained some good students along the way. And that using those laboratory-based measures, those granular measures, we're able to detect effects that global standardized measures often will miss. So please keep that in mind when you see newspaper clippings about claims. Acknowledgements to everybody who was part of this over the time, especially support from NICHD, including our newly renewed U54. And thank you for listening. The Lifespan Institute is a great place to work. It's much like the environment here, very collaborative, very integrative, very interdisciplinary. Thanks for your attention today. Thanks.
A comment and then a question. I think the comment is just to reinforce how important your careful psychometric work is on these process measures. I think, as you know, the NICHD is now investing a lot of effort because clinical trials in other populations with drugs have failed, oftentimes because we rely on these kind of global standardized tests. And so you really have kind of this model of how we should be working in the field. And
Thanks.
so I really want to reinforce that. And then, so here's the question is, so in these clinical trials, obviously everyone gets kind of the same amount. I mean, have you begun to think about more individualized treatments, either based on kind of, if we know enough about the genetics, so we know about metabolism of the LC-PUFAs, which is a fun word to say, or environmental behaviors and things like that that are going to affect other aspects that would affect that?
Yes. In fact, so the question is, there's like 4 questions in there. And as you tick them off, I think I have answers to many of them. We're really interested in whether or not the kinds of things we're seeing in terms of these effects being stronger in low-income populations or low SES populations is real. And so so in other words, in the grant we have now pending, we propose to oversample, for example, Hispanic populations in the Kansas City area, which is there's a lot there that we be able to see, and particularly that group we think is very deficient. So that's one piece of it. The genetic piece, we actually have data. There is a SNP, a single nucleotide protein of fatty acid desaturase. It's on the 11th chromosome. And there are two genes for this. We can evaluate that, and it's an interesting. I don't have the data with me, but I can kind of explain. If you have the 2 minor alleles, if you're missing the gene for fatty acid desaturase, you can't metabolize the DHA. You can't metabolize the fatty acids, the omega-3s. What happens is that if you do have that, you're 25% deficient in DHA. So we have that on the moms from the Kudos. We thought, great, let's supplement them, and we'll get them up to normal. And in fact, if you supplement them, they come up, they're indistinguishable from moms who have both alleles or one major allele. But what's really interesting is if you supplement them on DHA, they come up to normal there, but their other lipid profile drops, arachidonic acid drops. And the reason why is because all of these fatty acids, and I didn't show you the metabolome, the metabolized, metabolic paths, is that they all compete for the same enzymes for metabolism. And so when you increase DHA, in these moms who don't have the gene for that, it stresses the system and their ARA drops. So it's really an interesting gene by environment interaction that if you try to just simply, you're not able just to replete them very easily. And then when ARA drops, you have all sorts of other things that could go wrong. In fact, we think that's what happened in that very high dose. We basically overdosed the kid. That's the wrong word for that. But we gave them too much, and we basically got the DHA ARA at a balance in that very high dose. And that's why right now the suggestion is you don't exceed 1-to-1 or 2-to-1. So that's it. But we are interested in these other things. Oh, other things that are happening at home. We take diets. We take data on diets. The diet, everybody in Kudos got DHA later on, and that didn't seem to vary as a function of the group assignment. In the Diamond study, nobody got DHA later on. Their diets were uniformly poor, and I don't even want to talk about it. But it certainly didn't affect them. So.
As I understand, it's incredibly hard to get permission to run studies on mothers and infants. How does that work in compared to a normal trial with adult population?
I didn't hear the first part of it.
How does it work to get permission to conduct studies on infants and mothers as opposed to normal adult populations? Is it much more difficult?
Well, what we're doing is providing a potential benefit for a lot of these studies. We're providing a potential benefit by adding DHA into their diet. We'd give it to them for free. And so getting permission from an IRB is not that difficult, frankly, because we are benefiting them. I will say, though, that the criterion for that is changed over the years. So, for example, in Kudos, we gave them 0. You couldn't do that anymore. So that Kudos was funded in 2004, at a time when moms were not routinely getting DHA in their diet, in their prenatal vitamins. Now they are. So when we went back in to do the next study, which is a higher dose, we couldn't contrast it against 0. We had to contrast it against 250, which is the normal thing. So the boundaries are changing. The criteria are changing for what's ethical or not. For example, Diamond, those babies got no LC-PUFAs. We could not do that study again today, and because that wouldn't be ethical, based on what we know now. So I hope that answers your question.
Thank you.
People have been using the Bailey scales for decades.
Yep.
What's the justification? Is there any predictive validity of this test?
Yes. No. Yes and no. It's predictive when you get out to about 18 months beyond when language kicks in. It's correlated about 0.5 from there on out. We're right at 18 months. It's right on the border of whether it's predictive or not. But it's just that it's an agglomeration of all sorts of domains in there. And so we think it just obscures. If the effect is more specific to attention, for example, which is what I think is happening, you won't see it on the Bailey, and that's what part of it is for. The other thing is the Bailey. If you go back to the history of the Bailey, it was designed as a screening, as a tool not to be used as an IQ test, but as a tool to identify kids who are at very high risk for later developmental delays. It's really good at that, and in fact, in the Domino study, the Australian study, they didn't find it as a function of their different doses of DHA. They didn't find any difference on the Bailey in terms of overall scores, but what they did find is that the high DHA group, actually far fewer of them ended up in a high-risk category. So in terms of classification, I think it's okay for that. In terms of identifying kids who are at high risk for delays later on, I think it's okay for that. Whether or not a child who scores 108 on the Bailey is different from a child who scores 100 I'm not sure that matters. So.
Hi there, I have two questions.
Mm-hmm.
Do you have any clinical trials on postnatal breastfeeding mothers that were supplemented? And my second question is, what is your requirements or recommendations on the actual 600 milligrams of DHA?
Yeah. We haven't done studies where moms were postnatally supplemented who were breastfeeding, but they have been done. The results are pretty much as you'd expect. If you supplement moms with DHA, for example, who are breastfeeding, you get higher DHA in their breast milk. I don't know that produces a huge benefit. I actually don't know whether that actually does improve the outcomes over and above breastfeeding. My guess is it doesn't necessarily, but so that would be one answer to the first question. The second question was recommendations. Right now, the daily recommendations for pregnant women is 250 milligrams. That may change as a result of these outcomes of these studies. We'd like to see, we contrasted 600 versus 0. The grant we have right now pending is 1,000 versus 250. So we'll be able to tell whether that's a best test to know whether or not that high dose is different, has benefit over and above what typically is recommended. So, yeah.
I've done an animal model looking at perinatal, so both during gestational and pre-weaning in animals, looking at DHA deficient versus using AIN 93G, which is a pretty standard modeling what we see in a typical situation where you see a lot more omega-6 than omega-3, but still adequate amount of DHA. And we saw extreme sociability deficits using the three-chamber social deficit approach that Dr. Crawley developed, as well as anxiety-like behaviors in the offspring, which was interesting that you showed the anxiety-like increased fussiness. As far as the questions, when you look at the blood profile, of the, do you do a full fatty acid analysis? Because it would be interesting to look at the levels of EPA as well, since some people have suggested EPA is just as important as DHA. Whether or not I believe that.
Yeah, that's a Susan question. And that's the danger when you're a psychologist and you go ahead and give a talk like this. Someone asks you a question, goes like, I know the answer. Susan knows. I don't think we did a full lipid. I think we just measured what we just measured the omega-3s and omega-6. We didn't get we just measured DHA and ARA, but we didn't. I don't think we got EPA on there. So,
okay.
Yeah.
And then one last question. Did you look at any sex differences in any of your studies?
Yeah, we include sex in all of the in all the analyses that we do, and it didn't come out. But it's really interesting that in some of the big clinical trials, there is when you break it out that way, you find an improved outcomes for girls relative to boys. The Australian trial did that. They find that, and if you talk to the fetal programming guys at Oregon, for example, Oregon State or Oregon Health Sciences, they actually have models for how the lipids would be differentially incorporated depending on the sex of the organism. So they really would strongly believe that. So I think in fact, our Australian colleagues ran into a little bit of trouble because of the outcomes of their studies. The hospital administration said, okay, well then we'll just give it to girls, which is not a very optimal situation. So, yeah.
The UC Davis Mind Institute was created in 1998 with a promise to find cures for neurodevelopmental disorders. Every day, our physicians and researchers come closer to fulfilling that promise. Their groundbreaking research on autism, Fragile X syndrome, chromosome 22q11.2 deletion syndrome, ADHD, and other brain disorders are helping children achieve their fullest potential. Please visit our website to find out more about current studies upcoming events, and how you can help make a difference.